So let's try again. Yeah, thank you very much. So Alzheimer's disease is the most frequent cause of dementia, affecting about 35 million worldwide. And the major pathology of Alzheimer's disease is beta amyloid, as you can see on the left in the histopathological staining, where amyloid plaques deposit outside the cell bodies. And histochemical analysis has done a staging of beta amyloid development throughout the different course of the disease, going from left to right on the upper row. And you see that amyloid emerges quite globally, most pronounced within the posterior medial parietal cortex, and then spreading only later to the medial temporal lobe. Pipette imaging and other traces with high affinity to beta amyloid have replicated in humans this histopathologically established sequence. A more clearer, more distinct spatial-temporal sequence of the development spreading of pathology is seen for tau. Neurofibrillary tangles are deposited inside the cell bodies, and Heiko Braak has established the temporal sequence of tau spreading throughout the brain, which first emerges within the entorhinal cortex and hippocampus area, then spreading to the connected medial frontal and posterior medial brain regions Jacob Vogel talked about, and eventually affecting the whole brain. In the last 5 to 10 years, PET tracers with affinity to tau, pathologic tau, have emerged and could basically replicate this spatial temporal sequence. Now, a big question is who's developing these kind of pathologies, and genetic factors have been identified. First of all, these mutations in the amyloid precursor protein, so APP, which you see here on the upper left axis, and within the presenilin, which causes Alzheimer's disease and are majorly responsible for the development of beta amyloid pathology. However, those mutations, as you may know, account only for 1 to 2% of all AD cases. The strongest risk factor of Alzheimer's disease is ApoE4 genotype, which regulates the clearance of beta amyloid from the brain. A new mutation that came up within the TREM2 gene here on the upper left is extremely rare, but associated to a similar degree as ApoE4 genotype. It's related to microglia activation. On the other hand, one genetic variant in the BIN gene is highly frequent and still relatively associated with high risk of Alzheimer's disease. However, the mechanism how this SNP is associated with Alzheimer's disease is not well known. Meta-analysis have specified that the risk factor, the odds ratio, is about 1.2 associated with this particular genetic variant of the BIN1 gene with a really high prevalence of 37% in the population. So it's really important to find out what the pathomechanism underlying this genetic variant is. BIN1 stands for bridging integrator 1, which is involved in clathrin-mediated endocytosis. And this property makes it a good candidate to be involved in the spreading of tau pathology. So during the recent years, more evidence has emerged suggesting that tau spreads in a prion-like manner throughout the different brain regions, which may explain this highly hierarchical, distinct spatial-temporal sequence of tau spreading. So tau is constitutively released from the cell body, where it's being taken up by the synapse, via endocytosis. So it spreads along the axon to the cell body, then is released and is taken up by the other neighboring cell or synapse. And this process of spreading is mediated by endocytosis, which you see on the lower left, which is regulated by dynamin. And BIN1 is actually binding to dynamin and regulates this clathrin related endocytosis. Higher BIN1, isoform 1, actually decreases the endocytosis. So a BIN1 deficiency may actually reduce tau spreading, because tau is being taken up within these vesicles and then transported throughout the axon. However, this evidence has only been suggested or shown by in vitro studies. So far there was no evidence in humans. How about the relationship between BIN1 and the other major pathology, amyloid? Because BIN1 is implicated in the uptake, so in the endocytosis, and this is important for the uptake of APP into the cell body, where it's actually cleaved by beta secretase. It might also play a role in amyloid generation. However, recent evidence in transgenic mouse models of Alzheimer's disease have shown negative findings. But still the question is, can this be also translated to humans? So in order to address this question, how BIN1 SNP is associated with amyloid and tau pathology in humans, we assessed subjects from the Alzheimer's Disease Neuroimaging Initiative, which is open to researchers. We had about 40 BIN1 carriers that were either cognitively normal or had mild cognitive impairment, and 49 non-carriers, which were also either cognitively normal or had mild cognitive impairment. They were pretty much comparable in terms of the basic demographics, except that, and you see this on the lower row, that the BIN1 SNP carriers had lower memory performance compared to the non-carriers, both in the cognitively normal group and in the MCI group. So next, we obtained tau within those Bragg stage-inspired ROIs, which have been mapped into the MNI space. So we obtained ROI tau PET values within each of these Bragg stage ROIs, and compared then the tau levels between the mutation carriers versus non-carriers. And you see that throughout each stage, the carriers, so the BIN1 risk carriers, had higher tau levels compared to the non-carriers here in yellow. How about beta amyloid? So we again mapped this or extracted beta amyloid PET within ROIs that corresponded to different amyloid stages. And here we did not find any difference at all, so highly consistent with preclinical data. Second of all, we were wondering whether the tau PET pattern has a spatial correspondence to the BIN1 gene expression in the brain, as assessed by the Allen Brain Atlas. On the left column, you see the group mean of tau PET. On the right column, the mean expression, and then the next two represent a mapping after thresholding. And when doing a spatial correlation between the tau PET uptake map and the gene expression map of BIN1, we see a significant correlation suggesting that there is a spatial overlap between BIN1 expression pattern and tau PET. So next we were wondering whether the association between BIN1 and lower memory can be actually explained by the association between BIN1 and tau PET. And so we did a mediation analysis and found that indeed the influence of the BIN1 SNP on memory performance can be explained, at least partially, by its association with higher tau PET. So in summary, the BIN1 SNP was associated with higher tau PET, but not beta amyloid. There seems to be a spatial correspondence between BIN1 gene expression and tau PET, which may hint to a spatial vulnerability to tau in these brain regions. And last of all, the effect of BIN1 on memory decline may be explained by association with tau PET. So in conclusion, considering these previous findings that BIN1 may be involved in the tau spreading, which you see here in the upper row, where the authors showed that tau spread along axons, infecting other neurons that were separated in different chambers. We proposed that the BIN1 SNP may regulate the endocytosis, and thus a dysfunction of BIN1 or a deficiency of BIN1 may enhance the spreading of tau in humans, which is then which entails cognitive decline. Thank you very much. And last but not least, of course, I would like to thank Nico Franzmeier, who I mentioned already, and my whole team, as well as my collaborators. And thank you for your attention.